Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology, studying. Oh, oh, sorry, studying the skin, studying Staph aureus by growing it on skin, infecting skin I grew with it. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based at NYC, dedicated to the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist since then, and I'm currently working as an editor for a scientific journal.
Every week we meet to speak about microbiology, and today it's our deep dive week where we're going to go figure by figure paper that we chose with input from Twitter, maybe like a month and a half ago.
Yeah, and usually every week we cover a swathe of published papers that have caught our interest, and at the end of the month we put them all together into a poll. And if you pay attention to at microtwjc, we'll be selecting papers, putting up a poll so you can vote on it. And if you have a paper you want us to look at, please tweet at us, and we'll see if we want to cover it, or if the the amorphous blob of Twitter wants to cover it.
Yeah, absolutely. You can see any of the papers that we discuss in our shared Zotero library, linked in the doobly-doo below. And specifically, I just want to point out that there's a folder called Potentials, and that's the one that has things up on the docket. And for our presentation today, it's under Background. Things are organized by date there, and we have the papers that we used for this presentation.
And we want to hear from you, so please use the comments or tweet us at microtwjc. So yeah, without further ado, let's get into our paper. Um, so our our story begins with just your average super genius plant scientist working on developing a biorestorative formula when some evildoer plants dynamite under his desk, and in the subsequent explosion combined with this formula, this scientist was transformed into the superhero that we know as the Swamp Thing. So
It's a weird
what?
superhero. like,
Yeah, it's a
just the
superhero.
most.
So you may think this is a weird way to start the paper, start our discussion on a scientific paper, but there is a point to this because the theory like that was brought up in one of the later theories. Like, how could this have happened? Well, the idea was that this plant restorative formula superpowered the plant. So this person wasn't transformed into the Swamp Thing. What happened was that the plants around him ate him, and then through that process absorbed his memories. And this was based on an actual idea that's put forward in the paper by Jason Woodrue, another character in the Swamp Thing, who's also known as he's a bad dude. One of the bad dudes. But anyway, I can't remember his name.
He's a
But
bad dude. I
yeah,
didn't realize. But he's
yeah,
a
yeah.
bad dude who knows the science behind the person he's fighting, I guess. That's
Yes.
it. Yeah.
So to quote him, the planarian worms are the key. You see, a while ago they taught a simple planarian worm to run a simple maze. They educated it, then they chopped it up and fed its remains to a batch of planarians who couldn't run the maze. Except by digesting their educated comrades, the worms could run the maze perfectly. So, this is weird comic book science fiction, right? No, never would have happened in real life, except these are based on actual experiments. Yeah, by pretty performed by Dr. James McConnell. Uh, this was done in the 1960s, and yeah, so they were published. So James McConnell ran his own scientific journal that was called, I think, Worm Runners Digest, where he published a number of results about this, and then they were reprinted in the Journal of Neuropsychiatry, and it's interesting because he was. I mean, this is an interesting story in itself that I could really go down. But we don't. We're trying to cover a paper that was published today. But the idea was that these worms were somehow figuring out the key to memory, and the
I
and
mean,
unfortunately, like it kind of
the
fell out of favor
experiment that
because
was
yeah,
I don't quite I didn't dive into this as much as you might have, but it's the same experiment though, right? That this James McConnell did, like exactly what Dr. Rudru said in the comic that they fed
yeah,
worms to
it
other
is
worms,
basically
and then those worms could do
yeah,
the things that the first worms were trained to. Yeah,
yeah,
except maybe
yeah. So
not
he
it wasn't replicated. It wasn't very well replicated in the literature.
it's weird because like trying to trace like this research down, it seems like there were one or two scientists who replicated, but then they tried to replicate it in mice and higher
Mm,
animals, and it didn't replicate at
mm,
all. So there, and I think some people did have difficulty replicating it, and there's a lot of controversy about this.
right.
And in the end, it fell out of fashion. And I mean, we'd love to dive deeper into this, but I'd need to contact a
Yeah.
neuroscientist who'd be able to
Yeah.
talk more about this with more authority. Um,
Yeah.
but yeah, I mean, if you want that, then then at us in the comments. Um,
So
but yeah,
yeah.
yeah, it fell out of favor. People follow up on it.
And so I guess that's a little bit of a segue into the paper that we're going to talk about today. There's a specific lab, this Murphy lab at Princeton, that is looking at something called transgenerational epigenetic inheritance, meaning that, I mean, I think it's exactly this idea that like the worms could somehow pass on their memories or something, right? Some sort of information through maybe not traditional genetics, but some sort of epigenetic things through all their different generations. And actually, I think if we go back to this Google thing, so like the reason why this is, I guess, an important or an interesting sort of thought is because there's this principle called the Weissman Barrier that says that information only moves from germline cells to somatic cells, and that somatic cells don't pass down any information to later generations. So that should make sense to most people because there used to be, if people are like, Darwin versus Lamarck or whatever, there was the thought that, oh, like you can't just like learn during your adult life, you can't pick up certain adaptations that then you transmit to your children, right? So I think something like that. Do you recall what this Lamarckian
Oh
example
yeah,
is?
I recall because in every genetics lesson when you're in your learning genetics for the first time, they'd all make fun of Lamarck, saying like, okay, they did this experiment. Well, Lamarck said that if there was like a giraffe, like one, it like kind of tried to get the tall leaves and then got disappointed, so its children were encouraged to get the tall leaves, and
Yeah.
then later,
Yeah,
like
it's
through
like the
genetics, it
stretching.
would
It's like
like,
the
yeah,
stretching aspect of your parents, right? Like trying to stretch up there to grab those leaves. That's like somehow getting transmitted to your children to then like help them stretch. So I mean, that's like I don't know if he ever used that specific example, but that's like the example that people always use in like classrooms to say like, oh, Lamarckian evolution doesn't make any sense. But I think now that people know that there are like epigenetic markers, that there is a thought that, oh, there is some things that might get transmitted through, but the Weissman barrier is still like an important concept here because like we come from, well, multicellular organisms, they often come from like this germline, right? These like sex gametes or whatever. And so like the DNA that makes up our whole body is derived from that germline cell, not from some other random cell, right? So you could imagine like if you're getting these like epigenetic marks or whatever that's changing your gene expression in a specific tissue, that how does that there's a natural problem that emerges.
yeah,
How does that tissue pass on, right, its epigenetic mark into the germline cell that's going? So like the Weissman barrier is saying that the unbroken lineage of our genetics is held as multicellular organisms is held in our germline. And having a phenomena like transgenerational epigenetic inheritance is sort of challenging, right? That notion, like, oh, actually, there are because we've seen this phenomena happen, right? Like we are like there must be some molecular mechanisms that are able to transmit the information from those specific tissues that are doing the thing to the germline in some way.
yeah, because there's only so much information you can cram into either an egg or like a
Mm-hmm.
sperm, and it's hard to figure like if my arm gets cut off, how do I transmit that information to my kids? Is that going is like DNA going to take a long journey down? So it's a complicated
Yeah,
problem,
yeah,
and
because
it's
yeah, like when your
yeah.
when your arm gets cut off, so like all those cells are getting wounded, right there. Maybe their genetics are changing, right? They're expressing different things because of that
Oh
wound.
yeah,
But that's not those cells are not the same cells that are going to give rise to your progeny. So yeah, this question emerges, this Weissman barrier, right? Like sort of principle is at play there, saying, well, how do you expect that information to even go through, right? And that this Weissman principle is more saying like it doesn't. But we can see evidence in this particular. We're going to see evidence in this particular experimental setup of that information transmitting, and so the question will be like why and how.
Yes.
Okay, so in the next
So
slide, these are the people that did the work. It's mostly done by this Murphy lab. They're both at Princeton, the Murphy lab and the Gatai lab. The Murphy lab works on C. elegans, which is the model organism that we'll be looking at. They mostly study longevity, and specifically maybe like epigenetic regulations of markers of longevity, something like that. So I think that that's what gives the interest into this paper. Specifically, the two first authors who are sharing equally in the work are, oh, I think I just realized I think I got the names wrong on it. That's
okay.
Yeah, I was just like looking at that picture. That's not. It's Rebecca Moore on the. It's Rebecca. Rachel is correct, and then that's Rebecca Moore. Right.
Okay.
I wrote Edith Blackman twice there.
Okay.
Yeah.
Oops.
That's okay.
Apologies if you're watching.
And so those are the people who did the work, and then the labs have postdocs in them. Some of those contributed, another graduate student, and then there's a second lab that did some of the work here. They seem to be more like maybe they're brought in because of this like viral transposon thing going on. They're more of like a cell biology, bacterial cell biology lab.
Yeah.
And then one person here is pulling through the bioinformatics.
Right. Yeah. So we've got this other slide coming.
Yeah. So So I just wanted to do this as like the history of like memory and like thinking about like how do we study memory in animals and in anything, right? And some of the earliest information came from this really old example of like somebody who lost part of their brain, right? They're called HM. That's like the, I guess, anonymized name for this person. But what was discovered is that while they lost part, you've probably heard the story. You lose part of your brain, you can still do a bunch of things, but they didn't have memory. And so that told the early researchers that there's some distinction, right, between memory and the other things that your brain does. Specifically, there's this distinction people make: declarative memory versus non-declarative memory. So declarative memory, I think we can understand, it's like, oh, I can remember this thing that happened to me, right? Like last week I went down the stairs, right? And like, or like every day I walk down the stairs and today I bumped into somebody down there, right? Like that memory, like I'm able to talk about it and like conceptualize that cognitively. But there's another type of memory, right, and that's the picture. That's what the picture on the left or the right is supposed to show is like this non-declarative memory. Like you also just like learn associations over time, right? Like if something bugs you, right? If like, oh, you always get bad service at this one place, right? Like maybe you never say it explicitly, like I'm not going back to that place because I'm getting bad service. You just like slowly stop going to that place. But that behavior change is influenced by your memory. Like if you lost all your memories, quote unquote, right, you would no longer have that preference for that place. Like that, you wouldn't have those like innate internalized, like whatever it is, behavior responses about going back to that place. And so in this experimental setup, which is used in humans, also in rabbits, they puff air into your eyeballs and then you blink, right? Because you don't want air in your eyeballs. And they can train people like classical conditioning style, like Pavlov's dog, right? Like make some cues, right, that like you start associating with that puff of air, and like that's a type of learning that's being is being done to you. And so that's specifically non-declarative, right? Because there's no specific memory that we're like dredging up in that instance. It's just this behavior, and that's the type of memory that is going to get studied in animals mostly because of the way that we interrogate animals. Like we don't have these like long form questionnaires to be able to ask some of their declarative things. There are other ways they can communicate with us, but in general, a lot of the stuff that will be, and definitely the stuff that we're talking about today is going to be non-declarative.
Yeah. So like just to break it down, non-declarative is stuff that you just like I guess because I guess I'm going from movies. Like whenever someone loses their memory, they can still talk. They can still use like. Is that kind of non-declar? I'm not a neuroscientist, so I don't know these
Yeah,
things
yeah,
fully.
yeah.
But
I'm not.
yeah,
So actually, I don't think it's so. Yeah, like they're the ability to talk, right? I guess you have to remember the words, right? You have to remember those
mm,
specific words. So that's like in symbols and stuff. So yeah, so non-declarative is I think it's purely that idea that
just reactions, I guess.
mm-hmm.
Yeah, like aversive stimulus. When you smell food, you drool, and you don't necessarily remember exactly. Yeah,
Yeah,
yeah, it's
but it
complicated.
is something that needs memory, right? I think that that's the important thing. Like
Yeah,
the type of memory that is going to get studied in this paper is not the type of memory that like you memorize for school or something like that, right? Like the type of memory that's being
yeah,
studied in this paper is it's still memory, right? Because if you lost all your memories, you wouldn't be able to do certain behaviors, you wouldn't be able to display certain preferences, even if they aren't something that is like just a fact that you recall. And I think that's why that's why I wanted to introduce people to this idea because like we are going to talk about right we title this viruses that transmit memory but like what memory is being transmitted these aren't like these aren't the memories of what you ate last week, right? But it might be something
right.
that is it could be a memory that is linked to that piece of information that's giving you like a certain preference versus something over another.
Yeah, I think that's an important distinction to make because what we're going to be looking is we're going to be looking at focusing mostly on worms like the planaria, but not quite the same. And assuming that that memory will work the same way as a higher organism might be like over egging it because I think when the planaria worm experiments like came out, they came out with a bunch of like hype saying that you can gain memories by eating people. You can get a professor burger. I think was one of the things
Yeah.
they said. It was like going to be this. I think that we can boost your intelligence, but it didn't really pan out that way because I think that this phenomenon we're looking at is going to mostly affect worms, and it's very hard to generalize that up to. And I think that this is going to be an interesting study because of
Yeah.
that because
but
it's going
but
to.
I don't even
I
want
think
to
for me,
I
yeah,
think it's important not to say like this is just because this non-declarative memory I think does exist in humans as well, right? Like humans can be classically
mm,
conditioned. That's one of the reasons I'm showing like the eye blink thing, right? Because like people
right,
who have deficiencies in memory, they can lose that learning that they accumulate, right? Like with the training, with the whatever the classical conditioning is, the buzzer sound that then makes you blink a little bit more because like every time the buzzer's gone, they've puffed you in the eyeball with a bit of air. If you lose certain systems in your memory, like you will lose that ability as well. So it does happen in humans. It's just not like scientists are like trying to right. It's all about like teasing apart like the different categories of things.
mm,
And like in this particular arena, like there's many different categories of memories. And this memory that is going to be studied here is not the type of memory that you would say like the memory of your grandmother's face, something like that.
right, okay.
Yeah,
So yeah,
so we're gonna study this in a model organism, specifically C. elegans, which is a nematode worm. Uh, they have a defined number of neurons, so 302 neurons or 383 in males. And that sounds like I don't know. Does that sound like a lot to you?
I mean, it's more than the fingers I have on my hand, so yeah, it's
Yeah,
quite a
yeah,
lot of
it is
nerves.
kind of a
But
lot, but it's not as much as like the amount of neurons in like the human brain, right? Like this magnitude is
right,
yeah, it's like this is I mean, I was thinking about like actually 300 is a lot of anything. Like even like smart like some candy or something like 302 pieces of it is quite a bit. But
I
yeah.
mean, yeah, like in terms of Facebook friends, like I do feel like when I get to that number, there are some people in my friend list who I remember, but I don't know how I know them or where I met them before.
Sufficient yeah,
So that's kind of
there's
the.
sufficient complication at this number. Um
I feel like someone could memorize all of them, but it'd be very difficult and it'd take a lot of time.
Yes, yes. So there's this defined number of neurons, and actually, through a lot of efforts in the research community, there's also like their connections are also really well defined. So they have like all this information about the circuit of these neurons. So you can imagine like it's a very it's a good model system to work in because you can look at very specific lesions or something, and you can say how that would affect like the whole network of neurons. Of course, as we touched on, it's not like a walk in the park to be able to understand behavior through the set of some 100 neurons. But yeah, so we have a picture of the C. elegans here. There's some staining of like some specific neurons that they have. In D, like that's like a transmission electron image. And then I guess maybe importantly to focus on is E. Those are, and then a close-up, those are spots of bacteria where they place the worms on top, and the worms eat bacteria. So they just like wiggle around and they eat different things.
And you can tell they like one set of bacteria more than the other, so they go to that one.
Yeah, well, actually, in this example, it's kind of this is the one the reason I pulled up this one was because the worms on the right, they have a single genetic mutation that forces
sorry, you
that
broke up
basically
there.
makes them clump up together.
Sorry, can you repeat?
The worms on the right, they have a single genetic mutation that makes them clump up into little clumps. They call it the social feeding phenotype. So the reason why I picked this image out to show people, and again to link this to the idea of why are they using C. elegans to study this, is because people really want to know like the one gene, the one molecule that leads to a certain behavior. And I mean that might sound excessively reductionist, but I think the path of like setting out that problem for yourself ends up revealing different questions that you might have that then fleshes out the picture a little bit better. So like we see this a lot in science, right? Like you choose, like you want to reduce it down to this one little thing, but along the way you find out there are many factors that end up influencing that particular mechanism.
So here they've got a gene that makes them want to eat together, basically.
Yeah,
Is that
exactly.
what's happening?
Single genetic lesion gives rise to a totally different behavior phenotype where they instead clump together and they eat in groups rather than just all.
Oh, don't tell me. Is this the Thanksgiving gene by any chance?
Yeah, I mean that's coming up for.
Yeah.
Yeah, and so I mean that's pretty fascinating, right? Like one genetic change gives rise to like a whole set of behaviors, and those are the types of questions you can ask in a system where you have like the full genome, you have the ability to make those really directed mutations, and they're full animals, and they can go and they can live their life, and we can see behaviors emerge from that.
Yes. So right. Yeah, these worms, they like to eat the bacteria. So I've got some. So yeah, they go around. They like to eat bacteria, but of course, not bacteria are good for them apparently. So in this study, we've got some graphs showing PA14. So that's Pseudomonas aeruginosa, which is a bacteria that. So these are survival curves. So essentially, when something reaches 100%, that means like 100% dead worms.
Yeah.
Which isn't good for the worms. So when they encounter Pseudomonas aeruginosa, the bacteria comes up and it makes the worms feel very, very ill and die.
Yeah. I think for me, what really jumped out to me during this paper, just from like the bacterial pathogenesis point of view, is that like they were doing this research to sort of show that you can study the things that kill worms, and that might tell you something about the things that kill plants and animals. Like the same virulence factors that are hurting worms are also hurting like more complex organisms. And that would be like an amazing, it's a model really, right? Like C. elegans as an infectious disease model. But yeah, like basically they don't like to eat things. There are things that cause sickness even in them, even though they eat them.
Yeah, exactly. There's McDonald's at midday and there's McDonald's 3 minutes after midnight. I mean,
yeah. I do think, actually, I think that actually might be the right analogy here, because it's like it's still food, right? Like they're still getting some sustenance from it. Like they're eating it, but yeah, sometimes it just doesn't turn out right. It's like, I mean, it's
sometimes you get that smell, you decide, oh, that seems like it's really good, and then afterwards you're like, that was a bad idea, I'm never going to do that again.
So you would imagine that in evolution, then it would be good to learn how to avoid these types of foods that make you feel not so good, because they can have like small exposure, but like too long of an exposure to them, like that's going to result in some not good things.
Right.
And so that's actually, and this particular phenomena, this I guess important choice that worms have to make in their feeding is the behavior that's going to be studied in this transgenerational epigenetic inheritance paper.
Yeah, so transgeneration means between generations. Epigenetics means that kind of stuff happens outside the genome. And inheritance means that thing that's passed on between mother to son,
Yeah,
son to daughter, etc. Um,
yeah.
yeah. So I'm gonna go into like how they study these because I think the first I don't know if there's something you wanted to talk about before or is there um
Yeah, we could talk about it now. I mean, I know. I think it
yeah,
fits.
okay, cool.
Like, uh, like here we're talking about the so as I said, like this desire for worms to avoid some food versus others is like what they're going to study, and in those experimental methods, they have to be able to reset the scenario, right, between the generations,
Yeah.
and it's also make sure that all the worms that are going through the experiments are essentially the same age because they come from like the same set of eggs, right? That all hatch at the same time.
Yeah.
Um,
And especially if you're studying behavior, you want to make sure that you don't have worms from the last generation stick around because then they'd have already memorize that behavior anyway and just
right.
survive long enough for the next experiment. So you got this life cycle now. C. elegans is a hermaphrodite, so it can produce like asexually and sexually, I think.
Yep.
Um, and yeah, so they have this moment where they have let the worms mate, and then the worms mate and produce eggs, and then they add a bit of bleach to remove all the worms, and then they get pure eggs, and then they can figure out like make sure that all the worms are the same age. And I think one of the things for these experiments is that they're very careful because they've got this idea that behavior is being inherited. They want to make sure the worms aren't being stressed out or they're not having to experience behaviors aren't that aren't good because then there's a worry that that might confound the experiment. So you have to make sure the worms aren't like overcrowded. You have to make sure that they have enough to eat. Uh, because if you've got the premise that kind of behavior is passed down generations, you want to make sure that that the baseline behavior is
Yes.
standardized. Oh, I've yeah, Danny, we've lost your camera feed. Are you okay?
Oh, really? It's still going for me. Oh, I see.
Okay,
Yeah, it's saying my computer is stressed out.
right. Okay.
Let's see.
Yeah. So that's the
Close the
basic,
thing.
like, yeah. So yeah, that's basically how an idiot's guide to how nematode worms are
Yeah,
kept
so like
alive for
if they have the
experiments.
first set of worms they get trained, right? They would have some behavior. They can kill off all of that generation that learned things. They would only have the eggs from those worms that learned whatever they needed to, and then the next generation would be able to you'd be able to test them and see whether or not they still had the same behaviors.
Yes, and then you can so in order to test what they have, they've got a worm quiz basically where they have two blots of bacteria, and they need to want to figure out like which ones the worms first go to. And so the worms can go to either or, and when they go, they've got a dose of this compound called sodium azide, which makes the worm stop moving and makes them effectively a bit
Is
sleepy.
it actually
Um,
sleepy? Because sodium azide, I know it's a preservative, right? Like it's not very
yeah,
healthy to ingest
I'm
any of
saying
it.
I'm saying sleepy to be nice, but yeah, they really get paralyzed in that
Yeah, okay.
area.
I just wanted
Uh,
to clarify for people.
yeah, yeah. So yeah, this is how they try and work out which bacteria the worms are having
Hmm.
preference to, and they have this paralyzing agent because they want to make sure they have their first
Yeah,
choice. Because if they change their mind, then if you look at different time points, you're going to find they different preferences potentially, and that could be a confounder,
right.
perhaps. Or if they like finish off eating one bit of bacteria and then they go to the other one. So this is just a way to make sure you get their first choice
Yeah.
counted.
What do we got next?
Next, we've got like the preference. So this is how you find preferences. So they they can like E. coli, and so they use OP50, I think, a strain of E. coli as their control. That's just a generic thing that worms like to eat. And then they don't like to eat Pseudomonas aeruginosa. And they use this equation called the choice index, which is a ratio to figure out which ones they prefer.
Yeah. I mean, that's just, as you said, they get paralyzed when they go into those two different regions. So it's simple, right? Like, it's just the ratio between who ended up in one versus the other.
Yeah. Uh, and then, so in previous work, they tested this on like a panel of different strains, right? So you've got, and the main thing they found was that they Pseudomonas aeruginosa was really not liked when it had,
Yeah,
and then
it's
they had like an
specifically
yeah,
like we showed before, like Pseudomonas aeruginosa can kill worms, and when they're given the choice for feeding between these two different things, like they do not want to feast on Pseudomonas aeruginosa. I thought what was really interesting is like if you then knock out the virulence in Pseudomonas aeruginosa, they will end up eating it, and one choice. So it's definitely like linked to this idea of like you could get sick if you eat this stuff.
okay, okay. Sorry, Danny, your computer's
Oh no.
gone off.
Was that all
So
garbled?
if you, came out a bit garbled. So I don't know, is everything okay on your end? Um, okay,
Yeah,
you're
okay, let me
back.
try. Uh, okay. Yeah, sorry, all I want to say is in this one is if you knock out those virulence factors, then they won't show that preference anymore. So it is something you could interpret this as being derived from the idea that you don't want to eat food that makes you sick.
Right. Yeah. I fully agree with that. And then the next set of graphs are looking at the learning index. So checking like how the choices change based on uh let me
Yeah, so I think so in this one they have another like slightly not as not also not so good for like worms also not good for worms bacteria Serratia marcians, and they show that when you expose the if you train the worms ahead of time, right? So like you expose them to something they don't like, they will avoid that thing, but they won't avoid other things that they don't like. So they would rather not eat either. But I
mm,
think the idea here is that the training is specific for the thing that they see first. So like in this set of experiments, they're given like a period of time where they're exposed to the pathogenic version, right? So that basically
mm,
they get the sense of like, yeah, this stuff makes you sick. Like you don't want to eat this. Then they're put into that choice experiment where they can choose between two things that make them sick. And they'll avoid the thing that they know makes them sick, and they'll go to this other thing that kind of still makes them sick, but they've never experienced that before, so they don't care.
right.
Yeah.
Oh right, I see.
Yeah,
Yeah.
this idea is about like is like avoidance versus going towards it, right? Like you can look at the choice sort of two ways. Is it that they just go to food that they like, or is it they actually avoid food they do not like? And so by training them on this like one pathogenic thing and then giving them two potentially pathogenic choices, you get that sense that it's aversion rather than attraction that's governing this choice.
Right. And amazingly, like once C. elegans learn to avoid like Pseudomonas, that is passed down for generations. So what we have are like several. So these each of these graphs in like describe one generation. So the first graph is like the first generation that actually learned. They're the first one that encountered Pseudomonas. The rest of the generations won't, but they'll still have that behavior.
Hmm.
Um, until five, where they return to normal, where that behavior like drops out of the population.
Yeah. So it's kind of I find that to me that's like such a this is the phenomena, right? This is TEI.
Yeah.
It's like very perplexing from sort of just like an overview of it. It's like obviously something's getting transmitted, but it also wears off after some time. Yeah,
Yeah, that's
very
yeah, it's
interesting.
fascinating. Like, because this memory of avoidance is being passed down, and the question is, how would that happen? That's bizarre. That goes against this like idea of the Weissman Barrier, and
Yeah.
like all the times in school where I was making fun of Lamarckian inheritance because I was a nerd and not didn't have any friends. Like that. Um, but yeah. So the question is like, how does it happen? Yeah. And so hold on, let's see whether uh yeah. So we've got
The lab, the Murphy lab, they
mm,
they published three papers, at least what we could find, three sort of
mm,
like papers in this general area where they want to get a bit further, right? First to identify this weird TEI thing that's going on, right? They have their experimental system, they have worm choice and all that stuff, and they want to know why. So one of the things that they track it down to is this DAF7 expression in a very specific neuron.
mm,
So um, so they get some gene expression in this one neuron that when they see that expression go up, that's indicative of them then learning to avoid, right, the specific foods that they were trained on. um, and actually it's like there are a whole bunch of things that these are all different genes in the genotype column here. Like these are all different genes that govern different types of behavior. They make mutants in all of them, and they see what do they, what behaviors do they show. So actually, you can learn without this particular. I don't know. It's not a neurotransmitter. It's a TGF. It's a cell signaling factor, I guess is what we can
mm.
say. It's made in neurons, but it's a cell signaling factor. It controls like the expression of a lot of different chemosensory receptors, and but it's not necessary to begin like for the first generation to avoid, but it is necessary for all subsequent generations to avoid the food,
Yeah. So that
the nuts.
so yeah, that's a great observation. So it affects the passing down of generations, but not the actual initial learning. So it does seem to have an
Yeah,
interesting function. Okay.
but it is in the neuron, right? Like,
Mm.
but upon exposure during the learning, that's when it gets turned on in that
Right.
neuron, right? So I mean, this is how it's gonna get real. Like they're trying to relate, right? Like I think this is maybe the big one of the big challenges of like doing these types of uh, these types of like sort of neurophysiological understanding of underlying behavior is that like the brain big adaptive network, right? Like they are dealing with things in different very specific contexts, right, for different reasons, and you can see gene expression in them change in different times, but what does that correlate to? And so like it takes a lot of work, and I think that's this is why it's a really key finding that I wanted to highlight from what was found before in these papers is that they do track down, right, a specific signal in the neuron at the time of learning that then impacts what goes on later with the transgenerational avoidance.
Yeah.
Yeah,
So
this is
yeah,
a
it DAF
picture of that
seven.
neuron.
Yeah.
One of the 304.
But yeah, it's important for chemosensory genes. So that's essentially like the human version of that would be
Yep.
smell. But once you get to a microbial level, it gets a lot more complex because you can do all sorts of other things. Because everything's a chemical when you get down to it, like RNA,
Yeah.
DNA, like don't you, so
Yeah, I think I
yeah.
was reading the
Uh,
chemotaxis that it's involved with is like amino acids, like some amino
yeah.
acid behavior. But again, like whatever's known is only like what's been tested in the past. So in some ways, like what we're seeing in this paper and this body of work is adding more functions into the ASI neuron, right? It's also important somehow in transmitting this sensory information into some sort of transgenerational inheritance mechanism. Oh yeah.
Right.
No, I was just gonna
Sorry.
say also it has modulation of immunity in it, which I thought was really interesting. Like, oh, that also
Yeah.
that sort of ties into the fact that we've been talking about, Pseudomonas doesn't want to eat this because it makes them sick, because it's like you're eating pathogens. So, I mean, that to me, I was like, well, that's really interesting. There's crosstalk between how your immune system and all of these like food behaviors. Yeah, ostensibly what's food foraging behavior.
Uh, yeah. And so I think this will come in quite useful for the next slide because I think what they found was that one of the ways that worms can differentiate between these two bacteria is that Pseudomonas produces a bunch of these. I guess I describe them as like siRNAs or like
Yeah, I think they
microRNAs,
just call
but
them small RNAs. Yeah,
small RNAs. Yeah, that makes more
they're just
sense. Yeah,
they're just
because
small bits. It's not really clear what they do in Pseudomonas. Did you get that Did you get that from?
I didn't. Yeah, I didn't get what they were doing in Pseudomonas. All I could get was, I think, yeah, yeah, I couldn't figure out what
There are a
they were
lot
doing. I
of
think
like
like,
in humans too. Like this is something that I think is just recently being uncovered. It's like there's a lot of these small RNAs that get made, right? Like transcription doesn't always get all the way to the end of things, and some things terminate sooner than you would expect to make any gene products. And so instead, you just get all
mm,
these RNAs, and RNAs can base pair to things, and like that might trigger certain signaling pathways. And so, yeah, there's definitely a lot of function to be of molecule.
sorry, when you say sorry, you blacked out there. So when you said a function of, sorry, a lot of functions that, uh,
There's no explicit functions like during trans the small RNAs, they're not translated, right? They're not messenger RNA. They're not being made
mm,
into proteins. So what do they? I think the question is usually what do they do? They can base pair to other things, and then that might trigger like a double-stranded recognition, and so that might be their role in different systems. But largely, right, it's made in a lot of different. In bacteria, in us, we have small RNAs. The functions we don't know necessarily what the functions are, and that's all I'm saying. Like there's a lot of opportunity, I think, to pin functions to this group of molecules.
right. So yeah, they published a paper previously about this
Yeah.
subject where they were looking at this sRNA, which is P11, and they found that it was quite similar to mammalian genes. So there are some genes like there's a gene called McCoylin, C61, TMEM57, MAC. So these genes, these neural genes that are very similar to it, which is interesting
Yeah,
because
I don't even like this is so strange because this must be like I, I don't see this being like something that Pseudomonas cares about. Like Pseudomonas, I would
mm,
expect doesn't have any feedback from whether or not the homology of this small RNA it has to one region of um, a neuronal gene, like whether or not that would impact Pseudomonas survival in any way. But for whatever reason, it is
yeah.
that homology. What they pin down is that in all the small RNAs Pseudomonas makes, there is one RNA that has homology. That when you remove the homology, it no longer induces these worms to avoid that food.
Yeah, and they link this, I think, a bit to RNAi transport. So some, so I think worms they interfering RNA system
Hmm.
to knock down certain like transposons or viruses, and they've got like various signal transporters that can transport RNA signals between like cells. So the idea was that those same transporters that usually do that are taking in this P11 siRNA, sorry, sRNA as if it's one of those.
Yeah.
Um, and yeah, and so they related to this Pee-wee pathway, which is such a delightfully named pathway. It's the idea that like every organism has retrotransposons, these like dead viruses that live in our genomes, and they don't do anything, but when they do, it's annoying. So most organisms want to keep that under control to stop those viruses being. And Pee-wee interacting RNA is essentially like that. It's supposed to control transposable elements, and other spurious repeat derived transcripts in the genome. Um, and I mean, I think I've got an idea of why this might be beneficial to bacteria. It all depends on whether you think the bacteria are whether they'd prefer to be not eaten by the worm or they'd prefer to be eaten by the worm. Because if they prefer not to be eaten by the worm, this is actually quite useful to them as a signal to ward the worms off.
Sure. Yeah. That that's true. I can see that.
Um,
I guess the other thing
so
to point out here is that but I'm not sure. Right, I think that this is not about the initial eating though. This is about the transgenerational avoidance, right? So
yeah,
they do, right. So like for example, let's see, one of these figures. Yeah. Okay.
they do.
In
Yeah.
E, they make the mutant, so now it has the P11 RNA, but it doesn't have the maculin, you know, complementarity, and
Mm,
it doesn't have any preference anymore. But they've already been trained. They already had to be trained, right, for this in this
mm,
particular assay, right? Like this assay is after training. Um, so like
right.
it's not about that initial eating, it's about like the preference afterwards. Mm, I guess it
Yeah.
doesn't, they don't actually. Oh yeah, they do P, and then in F they then track it out on the generations, right? It almost becomes more distinct. I don't know if that's actually a good thing to look at, but like it's happening at P0, yeah, and then in F1 and F2 it's happening still. Yeah, I don't know. Never mind. I was
Yeah.
gonna say that like it
So
doesn't affect P0, but it does affect P0. Yeah.
yeah, I mean, yeah, I guess like the analogy here is that the tree frog that gets eaten doesn't benefit much from it, but every other tree frog that predator meets afterwards
Yeah.
does.
Yeah. Yeah,
Kind of
exactly.
that.
Exactly. But that's ignoring the fact that this bacteria is has all these virulence factors. So maybe like it's but there's
Mm,
many examples of like you have virulence factors that are not actually meant to kill worms. Right? They're meant for some other
right.
reason.
Yeah.
They just happen to kill worms. And maybe this is like a huge discussion anyways about virulence factors, which is why I highlighted it just because like we are infectious disease folk, and so like it's interesting to think about like
Yeah.
what exactly are virulence factors for in the life of bacteria, especially when like they end up killing a whole bunch of things that aren't necessarily like the hosts, if you will, of these particular pathogens.
Yeah, you're absolutely right. I mean, this is a much bigger discussion because I mean, yeah, you always assume that you're the target of the virulence factor, but you might not be. You might be for some
Yeah,
other reason.
yeah. Okay, but let's move on. So I think this is where we are before this paper comes, before this paper that we want to
Yes.
discuss comes to us, right? And that is we understand that worms want to avoid eating this stuff, and there's a RNA inside of Pseudomonas that gets sensed by a chemosensory neuron ostensibly, right? That then allows the worms to avoid eating that when they're given the choice later. But more importantly, or just more interestingly, every generation afterwards, the worms also avoid.
Yeah, for 5 generations they
Yeah.
avoid this, and so we don't really know how that happens, and that's what this paper is going to delve into a bit more. So the paper we're delving into is horizontal and vertical transmission of transgenerational memories via the SIR1
Mm-hmm.
transposon. Yeah, and so let's go move on. So the first figure, I think they illustrate their training techniques. So they've got their E. coli controlled trained worms. They got their PA14 trained, and they got some E. coli, but they've got the RNA added to them. So these are not pathogenic E. coli. They just have that RNA that warns off the
Mm-hmm.
worms. So I mean, what they tend to find is that when the worms are just E. coli trained, initially they're attracted to Pseudomonas, but then if they're trained on Pseudomonas before then, or if they have the sRNA, they tend to not have to tend to be aversive. So this is the graph that shows that. So you got lawn trained, so they met the Pseudomonas,
Yep.
they had the stomach upset, and they don't like it. And then you've got the one with just a P11 siRNA,
Yeah.
where they just have the RNA, which is just the warning signal, and just that warning signal is enough to train them to be aversive to um, to the
Yes.
Pseudomonas. Right.
Yes. And this is stuff that we'd see in the previous work. They're just sort of recapping it in the first figure.
Yeah, always good to replicate just in
Mm-hmm.
case.
Mm-hmm.
And then the next step is where they take lysate. So lysate is basically you take a worm and you chop it up into very, very small pieces, and then you expose naive worms. So you just squirt out those. So effectively, you're feeding these previous generations worms to naive
Yes.
worms. So this is kind of replicating those McConnell experiments.
Mm-hmm. Mm-hmm. Yeah, instead of training using the bacteria, like the aversive bacteria or the signal from that aversive bacteria, let's train the worms using the worms that learned to avoid that signal.
Yeah. Um, so yeah, this time they're not. These are naive worms that are being fed off of the different generations of worms that have had that signal. And they have that same aversive behavior.
Mm-hmm.
So when they go up the top graph, they are avoiding. So these graphs are the choice index, and each dot is one of those choice experiments. And yeah, the higher they go, the more averse they are to the Pseudomonas.
Yes.
So,
And I think this falls in line. There's a good there's a good clue here that this relates back to the transgenerational inheritance because that F5 generation, right, that we know they don't have that inherited memory anymore, right? When their lysate is used to try to train the worms, there is no difference in the choice index. Yeah, that's observed.
yeah, and so we've got many generations of worms that are being ground up and fed to other worms. Um,
Yeah, yeah.
yeah,
It
and this
produced it.
again shows that same
Well,
effect.
this one it recapitulates the idea that
Yeah,
they learn for 5 generations. So like the lysate, you have to have the lysate from like one of the F1 or P0 to F4 generation, and then yeah, and then that effect lasts for 5 generations.
yes, that's right. Yeah, I see. Oh, yeah, I see what the difference is. So in this case, the initial training was just on lysate, and then each generation
Yes.
afterwards
Yeah.
was.
They see
I
whether or
see.
not they
Yes.
still exhibit that behavior. I think in the can we flip back to the last one? Yeah, in the last one, this is lysate from different generations. Yeah.
Yes. So okay. So yeah, the next one is again they are using uh, a different strain of C. elegans, I think, as well. So there are multiple like strains of C. elegans out there. We're going to delve a bit more deeper into
Hmm.
that. They they also test out different bacteria. So got Pseudomonas fluorescens and Serratia
Hmm.
marcescens,
Yeah.
and they find that that yeah the choices don't change. There isn't that memory
Yeah.
seen with
It's not a, and this was, we saw
PA14.
this in some of the earlier work too, right? It's not a specific avoid things that hurt you type signal that's being transmitted. It's a very specific signal. Avoid Pseudomonas aeruginosa, right? PA14 that create these virulence
Right.
factors, right? That has this P11 RNA. Yeah.
Yeah, and then we've got uh, one of the supplementary figures that is also kind of confirming the F
Is
the
this
generations
supplemental figure 2?
2. I believe this is figure S2.
Yeah.
So they've got another. They look at Sir1 knockouts. We're going to talk a bit more about
Hmm.
those later, but the and they don't show any preference. But we also see like the N2 preference changes, and again by
Yeah.
F generation F5 the
Yeah, that
preference is
lysate.
gone.
Yeah, I mean, they didn't. I guess they didn't. This is it's a supplement, right? This one, what they're trying to pick out is just they did test every lysate from P0 to F5.
Yeah,
They didn't like cherry pick anything. They checked all of them. They all work. F5 doesn't work anymore.
right. Uh, yeah, and now they look at the RNA because they know that a lot of this is dependent on the
Hmm.
P11 RNA for it
Hmm.
all to work. In A they've got like the lysate which produces this effect, and then they extract just the
Yep.
total RNA, and they didn't find anything happening with that, which is odd because we'd expect that that I mean, the idea I'd have had is like, okay, they're somehow passing the same RNA through the
Yeah, like
generations,
somehow like
but it
maybe
doesn't seem to be
P11
the
like
case.
sticks around inside of the generations, right? They have like a, I don't know, RNA sac or whatever, and like those RNAs are the right, like they're important. Like I saw this RNA
Yeah,
before, like let's give it to my progeny so it can have the same effect. But yeah, it's not from RNA. It doesn't seem like isolating the RNA from F2 isn't creating this effect.
yeah. So to like, kind of counter test that, they go, okay, what happened if you destroy all the RNA? Will the effect disappear, or will there be any effect if we got rid of all the RNA? And actually, if they destroyed all the RNA, there isn't much change in the
Yeah.
behavior. So it
So
doesn't
in
seem
the
it makes
lysate, right? In
it
the
seem like
lysate.
the
Yeah, in the
yeah, in the
lysate.
lysate. Yeah. So this isn't in the initial training. This is like when you're in the point of choking up the worms. So trying to find out the factor that's passing between the
Mm-hmm.
generations.
Mm-hmm. Yeah, so it's RNase resistant. So yeah, that's bizarre.
Yeah, that is unusual. And then they decide to take the lysate and separate it out into all these different fractions. So density fraction, you're essentially trying to separate everything up by
Mm.
weight. So they put something in a test tube, and whatever falls to the bottom tends to be in one fraction, and things that float at the top tend to be a
Yeah.
different fraction. So the heavier stuff ends
Yep.
up in the bottom, and lighter stuff ends
Yeah,
up at
and
the
this is a
top.
really common way to approach a problem biochemically, right? Like if you have a mysterious liquid, in this case worm juice, blended worm juice, right, and it
Hmm.
produces an interesting phenomena, then you can say, well, this is a complex mixture of many different things. I want to find the thing, right, that is doing this effect. If it exists, it might not exist, right? But like that's the intent. I want to find the thing. So you do this separation technique where you're getting different weights in different areas, and so hopefully you're going to get closer, right? You're discarding a lot of things and you're purifying out something. You still don't know what it is because you've just purified it based on some criteria that you pulled out of your right, like you pulled out thin air essentially, right? Density. It's
Yeah,
like maybe the density is going to be different in the thing that you're looking for.
yeah. It's like imagine you're trying to search for something on a map. You just divide it into four quadrants and find it, and like kind of hone in on an area
Mm,
each time. So so when it's like you're casting out a net, it isn't going to be necessarily definitive. You just have to find a way to kind of uh, get rid of some part. So work out which areas you
yeah.
don't have to search. So in this case, they separate out into fractions and go, okay, whatever is happening must be happening due to a specific thing. So let's just pull out, like separate out in all sorts of different ways and see where the
Yep.
thing that is
Yeah,
causing everything
and
is
they
happening.
get a great
And they
result. Like
found here that
fraction 6 is the
yeah,
fraction where the activity is. It doesn't exist in the other ones.
yeah. So something that's floating right at the bottom of the test tube is causing this
So pretty
effect. And what they yeah, it's pretty dense. And what they find is they look in that fraction, and they find these virus-like particles.
bizarre.
Yeah, like so that's how it and that's how this neurological paper edges into our wheelhouse of
Yes.
microbiology, because suddenly it's about viruses. Yeah. So this isn't necessarily a new thing. So in previous work, some researchers did find that C. elegans did produce virus-like trans particles, and these were part of the CER1 transposon, CER1 transposon. Uh, yeah, their function isn't actually known. They seem to notice that they kind of accumulate in these worms. Okay, Danny, your sound is completely gone. I'm so sorry. Oh God. Yeah,
My battery pack.
okay.
One second. Okay. How about now? Is it back?
Yeah, I can
Yes.
hear you. You're using a
Yeah,
different
I'm doing
microphone, I
using a
take
different
it.
microphone.
Okay.
Not as good.
I mean, we have to work with what we have to work with, I'm afraid.
I can step away for a moment and attach one more. 1 second.
Okay.
Okay. Let's see. Maybe.
Yeah, but this sounds much better, thanks.
Yeah. Okay. Excellent.
Yeah.
Okay. Yes. So, oh yeah. Like this was this 2012 paper. They wanted to tell a story about what does this weird SIR1 retrotransposon do, and the only function that they could sort of ascribe to it was that they saw a difference in like the the eggs at the egg viability at different temperatures, and then the amount of these weird capsids at those different temperatures. So they're like, oh, maybe it's like some strange thing where it like restricts the temperatures in which the eggs could develop at. But yeah, there's a lot of them, and they go to the eggs. I think that's one interest. That's like one of the interesting things that they're pointing to
Right.
in 2012. And again, that's what makes it very interesting. Probably when these researchers connected the dots, right? Like this is sort of like I think the amazing part of like this research is that they have this phenomena that is strange and they don't know what governs it, and then they're able to connect it to another phenomena that's being seen 8 years ago 9 years ago in the literature.
Yeah, that's amazing. It's like sometimes you have different pieces of the puzzle. You have to that sometimes you left with a jigsaw piece and you don't know where it fits for like a whole like,
Nearly a
nearly a
decade,
decade,
yeah.
and then
And I
suddenly
mean, even if that's like
it's
science in a
suddenly
nutshell, I think
yeah.
in many ways. Yeah.
Yeah. So then they take out fraction 6 and they test whether that is good enough to have that
Hmm.
transgenerational inheritance. So that memory of aversion that can be passed between generations. Um, and it does. Until of course generation 5, which where they just forget everything.
Yeah, it's
They
some
don't.
weird magic cutoff point. I don't actually. You know what I just thought about is like it'd be really funny. Did they repeat the out to generation 5 for every fraction of the lysate or every fraction 6, right? Like fraction 6 purified from F1, fraction 6 purified from F2, from F3, and from F4, and then take each of them out 5? Is it that like at that point it runs out, or is it
Yeah.
that you get like you make up the difference until you get to 5?
Yeah. I assume because it seems to always be so. Whenever they do the grinding of
Hmm.
these worms up, it always ends up going to 5. But when the transmission is happening between worms, it might be a bit different because I assume the way you're grinding, you're gonna be exposing them a different way. You're probably gonna enrich it for
Hmm.
the fraction. So if we have like if say theoretically like between generations the dose of virions within the eggs
Yeah.
goes down, it might not necessarily show up in the way this
Right.
experiment is
But
done.
that's what
So
I think like that's part of the hypothesis, right? Like if you think that it's in something
yeah,
about like the dose of the virions, I don't think they ever like try to attribute it specifically to that, but like then you would
right.
expect to see that idea where like if you took up F4 and you made lysate from them and tried to train things, right, you'd get less of the effect like in the later generations because you're working from a smaller amount. But that's not might not be how it works, and I don't know whether or not that experiment also
Yeah.
sufficiently like excludes like whatever, yeah, the idea of dose for the virus.
Yeah, I guess it depends on like how you do that experiment because I guess if there's something like functionally changing in the structure of these like variants between the generation,
Mm-hmm.
then I would imagine that that the generation that gets passed down would matter. But if it's something else, this is yeah, it's really
Yeah.
all new to
Yeah,
me,
and I mean
and
it's all
yeah,
just like a weird it's like I'm just this is like taking the things that I've learned from reading this paper and trying to combine them into like a viable experiment that could be done, right? What would that experiment tell us if if it did happen, right? And what can't it tell us? That's sort of that's like a good way to interact with papers sometimes, and to sort of test whether or not you're absorbing some of the bits from it.
Yeah, so I guess an experiment you could do is take from each fraction and try to enumerate the number of these these varying virus-like particles in each one to see whether there's a difference. You could also try to do serial dilution to see whether that changes the effect that you get with when you feed different ones of them.
Yeah.
Or yeah. So I mean, the thing is, like, each dot is a choice assay, so we're looking at hundreds of experiments.
Yeah, to find out a difference, so this is a lot.
So yeah, it could be they're looking at these already, but it's quite clear this takes a lot of time and manpower to do.
All right.
I mean, yeah. So
let's see what we got next. Oh,
yeah,
repeating the same stuff just with this fraction 6.
Yep. And again, they're testing it with the RNAs to make sure that this RNA independent. So it's not dependent on RNA. Even if they take out all the RNA, it still happens.
It could be RNA inside of these viral-like particles, right?
Ooh, yeah, that is true.
They did earlier. They did like in like the previous control where they did. I think they did total RNA. Like they did some sort of extraction procedure and put that through. But like extraction procedure is also going to change the shape sometimes of RNA, right? And sometimes shape is really important. So I still think like that can't fully be ruled out. Like it's not RNA inside of a viral-like particle that isn't a specific conformation.
Yeah. Speaking of that, they actually look at the virus-like particles and check whether there's RNA inside them, and they do seem to find RNA inside them. And so this chromatogram just shows that.
And that makes sense because go to the next slide. I think it's perfect. That makes sense because what these viral-like particles are is they're retrotransposons. So they're from some old retrovirus, and retroviruses have a genome that's made of RNA.
Yeah, and most of them have like GAG, POL, and ENV that's like part of their structure of their genomes, and this research they found that like SIR1 has those 3 genes, so it could potentially, and they find it does create these virus-like particles.
This GAG POL, this is like the structural protein and the polymerase all together. It's made as a big polyprotein. It gets processed later. It's just like I think it's the way that you can say that this is probably from a retrovirus, because it's from evolution. Like this whole family of viruses have this very similar structure. And I do want to say that this retrovirus isn't a full virus, right? It doesn't have all of the elements that it would necessarily need. It's what's considered like it's a retrotransposon, right? So it's being integrated into the genome, and through some evolutionary time has lost some other elements that make it a fully functional virus.
Yeah, so it can only be like made in this organism and under certain conditions, so it can't infect other. That's why it's a virus-like particle, because it can't be
that we know of. I guess we don't really know. I think virus-like because it just doesn't have the rest of the things. Like, where's the reverse, right? There's no reverse transcriptase here. If it was a true retrovirus, it would need a reverse transcriptase because it would need to make its RNA into DNA, and then that DNA would get integrated somewhere else. Like, that's not what's happening here, but it still has these structural proteins and a polymerase.
Yes. And they do some microscopy to see that. So they create a SIR1 mutant that doesn't have this, and they also create a wild type. And you can see, like, in the wild type, you've got all these like green dots that are the SIR1 containing viruses.
Is that on
Apologies,
your
can you
side? Honking?
Yeah, that's on my side. Apologies. My next door neighbors have a car alarm. I'll repeat. Okay, right. I'll repeat what you just said. But okay, so what we have is they created a SIR1 mutant and a wild type, and they stained for SIR1. Those are those green dots that you can see on that microscope.
And the histone H3, again, that's a epigenetic marker for like some sort of histone conformation, and it's turned on eggs. So what you're seeing there is like the eggs, the developing
Yeah.
eggs of C. elegans. And as we said before in previous work, they showed that those SIR1 particles are important in this egg development process. They travel down microtubules and they associate with those developing eggs. So yeah, they have a SIR1 mutant and
Yeah.
doesn't make the particles anymore.
Yeah, and you can see in some of these particles that with the eggs there is that those SIR1 particles just seem to be associating with. I think 3 of them in that shot. So
which is so
yeah.
It's like, I think this is kind of crazy, but also not so crazy because I'm not sure if anyone's familiar with the story that the placenta has sort of fusion proteins, viral fusion proteins, are thought to have been like are the evolutionary precursor to some of the placental fusion proteins. So like, there's always ways in which weird pieces, relics of DNA that get left in our genome get repurposed for biological processes. And I think that that's the thinking around SIR1, is that this is something that was like laid into the genome of C. elegans, and now it's undergoing evolution for C. elegans.
Yeah, completely. And so they do some more confirmations. They take the lysate and they check to see whether the gag protein from SIR1 is there, and so they test all the fractions, and you can see like in fraction 6 they've got it there, and they got a couple of other controls, and and they got their SIR1 mutant to confirm that it doesn't have that. So there we go. And now they test out a couple of knockouts. So this is a key part of biology, of biochemistry, I guess, where you knock out a gene to see how whether it changes behavior or not.
Well, no, this is like they did their biochemistry, but now they're going to verify it in genetics.
Yeah, that's right. So yeah, we got your P0 SIR1. So these are the training things. So can these worms still get trained to get averse to the PA14? And find that yes, they do. But can that be passed down the generations? So with the knockout, it doesn't seem like it is passed down very well.
Yeah, so again, that's like, this delay in the worm behaving in this certain way, that's super interesting to me. And you have to remember, before we talked about DAF7, like it's also linked to a very specific thing that happens in the neuron. Right, so it's like both. It requires a signal in the neuron. It requires this viral-like particle. What's going on?
Yeah, exactly. And then they do RNAi, which is a knockdown, which is
this is like a second method, almost a second genetic method. Instead of making the change in the genome, they're making it at the translation level. Like, they just want to make sure that the messenger RNA doesn't even get translated. So SIR1 can't be made, I guess.
Yeah, I think that's right.
SIR1's still in the genome, but it's not being made. The viral-like particles aren't being made.
Yeah, so if the SIR1 RNA was doing something on its own, then this wouldn't if the SIR-RNA was doing something on its own, then you'd see that this wouldn't have an effect. Oh god, my next door neighbors are being
That's fine. It's like all sorts of weird sounds this morning for us. I'm also thinking like the internet might be like off for me because like the New York marathon is happening right now, and it runs right outside of my house. So there may be crowds of people with cell phones all trying to get internet connection outside of my house.
Oh dear. Yeah, but anyway, this RNAi confirms what we've seen previously and indicates that, okay, well, SIR1 has to be fully translated and formed into protein in order to have its effect.
And I'm sure that what they would see in these mutants is, like, if they did the fractionation and they looked at under the microscope, they wouldn't see those particles, right? Like, that's the mechanism in which they're disrupting this. No more viral-like particles are being made if you remove SIR1 in any of these different fashions. But they never show that directly.
Yeah. And again, we got let's see, we've got this. Oh, this is basically the same, but in a wild type strain of.
No. In this one, they're training with P11 now. So So like
Oh,
in all
right.
of those examples, right, they were
Yeah.
training with like the lysate or whatever. Right. Actually, we can go back. They should probably say what they were using in the last.
Yeah.
They were training on the
Yeah.
bacteria actually in PA14.
Yeah.
So they also make sure that this effect happens when they train with just P11 itself, the RNA.
Yeah. So, yep. Okay, and they find the
Mhm.
same effect happens. So they're dotting their I's, crossing their T's, getting
Yeah.
everything cleared up. And now
Yeah. So then
this one is
they have SIR4, which is like there's another like retrotransposon that sits inside of the genome, and so there's this alternate thought that it could also be SIR4 that's making these things. So again, they do it trained on lons in A and B and trained on P11 in C and D, but there's no effect when they remove SIR4 by RNAi. So SIR4 is not contributing.
I think this is a sensible experiment because so far they've seen like virus-like particles under the microscope, and they know it's a gag gene, but they don't necessarily know which SIR it belongs to. So they have to make sure because otherwise it could be a different virus entirely. So they need to kind of rule that out.
Absolutely. I mean, this is I think this is the essence of a lot of as we go through papers, right? Like in depth, we'll always see like what are the ways in which they rule out different possibilities, and then how do they show directly, right, what they think is happening. And the balance between the two is what makes for good evidence in a paper. Recognizing that it's hard to be 100% in any arena because all of this is work, right? Like every dot here was like time spent at the bench setting things up and like counting, right, for that dot.
Yeah, exactly. Yeah, and we've got
Yes.
ASI intensity. So this is looking at DAF7, which we talked about earlier in the show.
So they're gonna show that when they have the SIR1 knockout, it no longer has that distinctive upregulation of DAF7 in the ASI neuron. And remember, like this neuron is being lit up during that learning event, but the effect that it has is for later. It's for the transgenerational inheritance. So yeah, what about that?
Yeah, interesting. So when you knock out SIR1, this DAF it stops lighting up. It stops being activated when it reaches P11. So it implies that SIR1 is kind of upstream of that. It's happening before DAF7 gets activated.
Yes.
Right. Next one we've got yeah. So this is
figure 4A and B.
4A and B.
So they've done training with P11 and they've done training with PA4, but now what about if they do training with the lysate from worms trained on P11?
Right, yeah, of course. This is again like so they're looking at the lysate trained and fraction 6 trained, and they find that when they get rid of the SIR1, that any effects that they would usually see are gone. Yeah. The aversion is not there. So it's not. Yeah. So it's not just
again. It's like it's one more It's one more removed because it's like the worms trained on P11 that are now lysed are also affected by SIR1.
Yeah, so it's not just that it can't be someone like we've seen previously. Someone can't be passed down generations. Now deleting it shows that we can't pass on between different worms. So when the worms eat other worms that don't have the SIR1, they don't get that memory. Yeah. And again, they also. Oh, this is interesting. They also look at GLP-1. So GLP-1 is a gene that's involved in forming these granules in the germline. So I think that germline granules that contain RNA and other things that are transferred between generations, and and so knocking out GLP-1, I think, gets rid of that. So
they don't make their eggs in the same way, and it's also disrupting this action.
Yeah,
and that feeds back into this story. Like it's something about SIR1 during the development of the eggs, right? That is transmitting the information.
Yeah, so I think GLP-1 is involved in like meiosis in C. elegans.
Hmm.
But yeah, so it's implying that there is some effect in the germline as well. The so the so if you're going to try to hypothesize of how it's being how this is breaking the Weissman barrier, the idea would be that okay, so maybe it's being these the so on is going into the germline cells, but via
Yeah,
means that we wouldn't
yeah.
normally expect.
Exactly, exactly. And I think that we were trying to set that up like earlier during the discussion of this paper, right? Where we're saying that like, yeah, if you get your arm cut off or whatever, right? Those cells there experience something, but
Yeah,
how does that get transmitted? Well, the Weissman Barrier would say it has to get transmitted to the germline, and then this information also sort of says the same thing. So what seems like something that is going against it is actually it still goes back to that principle, right? That like it's the germline that's sending this information forward.
yeah, it's just not operating through the DNA or means that we'd usually ascribe to it. So, yeah,
but
that's
but what we'll say is doing a mutant, like showing that information via a mutant, is something that is many layers removed from the direct observation of what's
mm,
actually happening, right? So like, because who knows, maybe GLP-1 also affects other mechanisms that we just don't know right now. And so like then, right, like yes,
yeah,
the easy way to explain that mutant is that something in the egg formation step is being disrupted, but we don't know what other things could be disrupted by that mutation.
yeah. So they break this down into a model to try and figure out like where the things are being where SIR1 is being activated. What
Yeah,
stage is being important?
at what step, yeah, during at what step, because like this is like similar to the DAF7, that early DAF7 experiment where it's like they does it affect the initial learning, does it affect like the next generation, or is it the transgenerational inheritance? And similarly they have three in here, they're giving us three sort of theoretical ways that this could go down.
Mm,
And so they're going to they're going to give the RNAi for SIR1 just at the F1 stage, right? In the first
right.
after the generation has learned something, right? And then they're gonna knock down SIR1 in the first generation that comes out, and then they're gonna look at all subsequent generations to see whether or not they're passed down the information. And they have three different scenarios that could come out, right? One, that it SIR1 is important in the initiation of this phenotype. And so all that matters is SIR1 being present in that P0 generation. And so if you give RNAi in the F1 generation, nothing would affect, right? There would be no
Mm,
effect because SIR1's only important in that particular phase. And so what you would see is every generation is fine until 5. Basically the same thing we've seen at all other points.
right.
Um, but if it's for like the maintenance of this, so like basically something happens in that first generation, right? And if you break the chain by breaking SIR1, right, it's no longer able to pass down that information, then what you would see is the pattern that we see in orange or yellow,
Right.
orange, where P1 still gets the learning. Well, of course, it got trained, but then in F1, right, you lose it, and then because the chain is broken, it's no longer able to go.
Mm,
And then finally, the third scenario in green is what if it just affects that one generation, right? That it's the thing that's being propagated is somewhere else in the system, right? And SIR1 is just important, to make the DAF7 go, to engage, right? To engage in that specific generation the right mechanisms to change the behavior. So then you'd only see it in the generation that was affected with RNAi.
right. Yeah. So they're going to test these hypotheses in the next few
Mm-hmm.
next few slides.
Mm-hmm. Mm-hmm.
So let's see what if we get RNAi at F1 and looking at I think they're looking at different genes.
Yeah,
So
and they're going to look at even more than just SIR1. They're going to look at all sorts of genes that could be important in this process.
yeah, SID2, if I recall, is RNA interference transporter gene. Um,
Mhm.
so thought to be important during the initiation, I think. of this. So the initial like transfer. So I think the bit where
In
they
some ways, it's like a positive control because
yeah
they have already their prediction that they know that if you can't absorb, because we know that this one is involved in initiation, like to take up the P11, you said this before, right? Like they have to be able to eat it and then transmit it through their gut. And if they can't do that, then nothing will be learned, right? Essentially, you've blocked the early learning stage.
yes. And then they got PRG1, which is involved in the propagate. The idea is might be involved in the propagation, transmission to progeny. So I think we're looking at main ones we're looking at are the orange
Oh,
and
wait.
yellow lines,
Sorry,
right?
but this is. Oh, no, I just want to say they get. Oh, right, yeah. But because they're giving the SID1 in the F1 stage, right?
Right.
It doesn't block anything because they've already learned their lesson.
Yeah. So
Yeah.
so the choice index stays above all the time, except you're looking at the control, which is in grey. That's going to be hovering on the bottom because they don't learn anything because they were never trained.
Right.
Um. So yeah.
And then PERG1.
And PERG1 in the P0 is fine, but then when it's the F1 has the RNAi, that seems to be knocked down, like we'd see in like hypothesis 2. So maintenance propagation in the middle bit, then we see that okay, PERG1 might be involved in the maintenance and propagation of this behavior.
Yeah, and this is true because this is also something that I think they showed before. They were showing dependence on that Pee-wee Argonaut thing.
Yeah.
Later on, so like this was known that like it's something about how that RNA is processed during the transgenerational in F1 that sets up the memory to continue on through the 4 generations that it's supposed to. And so here it does break it, right? When they give the RNAi at that early stage.
Yeah, and then we see DAF7, which again operates green, so we know that happens downstream of SIR1, and we see here that that knocking it down with RNAi suppresses that behavior for the F1 generation, but then afterwards all its offspring have that avoidance behavior. So the information is there, it's just not getting acted upon.
Yeah, that's super strange. Yeah. And then finally, I guess what we're curious about, right, is SIR1. How they want to add SIR1 into the model,
right?
Right, and it also behaves like DAF7 in that it just acts in that one generation that it's knocked down on. It prevents any of the effect, but something is still getting passed on to the later generation. So once SIR1 is able to act again, those generations avoid Pseudomonas again.
Yeah, I mean, I'll be honest. The thing that I mean, I don't. Maybe they note, but yeah, the controls look kind of. It looks like they recycled the controls a little bit for this experiment because the controls look very odd as well. Because, because
you think they recycled. Go back to.
So for the first two, it doesn't look like that has happened too much because I'm looking at the yellow and grey lines to see how different they are, and they do seem to be quite different in B and C. But when we get to D and E, they look very very similar. I'm not sure what's going on there. But yeah, I would be concerned that might be a confounder in the interpretations, but it's a bit of a weird thing that I've only spotted because I've got this like now on my giant HD TV. I'm looking at it, I'm like, oh wow, that does look similar.
It's funny because we cut out the supplemental figure where they expand these. They expand each of these. I'm just gonna go down to it. And I'm gonna go down to it in the paper and see if there's some insight there. On that one, yeah, it looks like the control is the same across all of them, which is even odder. Is the scale different between A and B?
No, it's not.
Or A and C and D?
Yeah,
it's not. It's the same scale.
It's the same scale for all of them, and yeah, it's also. I mean, this is kind of, and I think like so for 3 of them the controls are very very similar. which is not something I. So actually now looking at all, yeah. So the only other controls are different is like SID2, and all the other ones have very similar controls. So that is,
Yeah.
um,
I think it's something in the data visualization because in the supplemental um, supplemental figure 5 from A, B, C, and D, looking at the controls there, P0 they all look different in the jittered plot. So like it's just the lines that look the same, but like when you break up the right, because each line is an
right.
average of the choice index from many different experiments. When you look at what they jittered on there, those
Yeah.
oh two of them do look different. I don't know, it's actually really weird. Like the DAF7, the PRG1 look the same. There's something strange going on, I think. Also, we're looking at the preprint. I wonder if this might have been smoothed out,
Yeah,
like
I
in
mean, I
the
did look at the yeah.
in.
I've got the full article here, and it doesn't look like it was smoothed out. It looks like so. I don't know what's necessarily going on there. I mean, it isn't necessarily a bad thing to reuse your controls if you don't have resources or if so. It's not, it doesn't necessarily, but it does it does raise questions if because you want to make sure that any effects on the day don't impact things. So if you're working with behavior,
Right.
so it could be that all those three experiments were done on the exact same day and had the exact same conditions, which would basically save resources. So that's why you'd see something like this. Um, and
Mm-hmm.
I need to check because I think they might have just declared that in their. I think they might have declared that in their figure legend. Uh, but anyway, yeah. So it's not unusual. It's just, well, it's just something that stood out to me a little bit.
Yeah, seeing the figures in blown up form instead of in the little Google Doc you've
Yeah,
been working out of.
sometimes that happens. Sometimes I mean, I found that a lot. Like sometimes you print something out and suddenly you see stuff that you don't see before. I don't know what the psychological effect for that is, but
Hmm.
yeah. Um, yeah. So next
So
one
let's see what
is
we got.
looking at the RNAi
Mm.
and the ASI.
They only gave it, yeah, so like typically they've been giving this RNAi like at the when they hatch and they're in their larval stage, but here they're trying to give it a little bit later to see if it has an effect, right after the worms have developed more,
Right.
trying to get a sense of like if it matters in the life cycle when they give it. So like not just like the generations, right? In every generation the worm goes through like a whole life cycle and so they're going to now like target the RNAi like only later in the life cycle, but it still seems to have it has the effect.
Yeah, and yeah,
Yeah,
they also look at the intensity of DAF expression as well,
yeah,
and they find that yeah,
yeah.
with the RNA, yeah,
It's not something specific. Giving the RNAi like even later when the worms are developed, it's still able to suppress this DAF7 in the ASI neuron, and yeah, so then have the effect of changing the choice.
yes, and then we've got so yeah, they look at suppression throughout the whole life of the nematode worm of C. elegans. Um,
Mm-hmm. Yeah, so you can see here like they're again just trying to give the SIR1 RNAi at very specific times, right? Like, do they give them at P0 for the whole life, right? Or just at P0 during the adult time, or both. And they don't when they just give it at that P0, or yeah, if they only give it during P0, then they don't get the effect in F1.
yeah, and
Right? Like those, they still have a choice difference between them.
yeah, and yeah, I think I've broken my eye slightly because I've just realised that the controls here have been kind of reused a couple of times as well. Uh, I
Now you can only see
I
reused
mean for
controls.
B, C, and D, it looks like those first
Yeah.
two have got a lot of very. So I mean, it's again, it's if they're all done on the same day, I can imagine why they did it because they're all because that saves time and it means that you can do multiple experiments at the same time. Sometimes now it just stands
Right.
out to me. That
Yeah, I mean, in this one, this one actually, I see this one definitely being done all at the
Yeah.
same time. The last one, I just don't know because some look similar, but some didn't. That's what I didn't understand why some were
Well,
similar and
yeah.
some
I
weren't in the
mean,
last one.
yeah. Here I
Yeah.
can sort of understand why the P0 would be like different because P0 is literally on a different day.
Yeah.
But then and F1 I can understand why they're all like very similar because again I can imagine them being done on a different day. But yeah, for the
Hmm.
DAFs for these ones where they've got multiple like generations, I'm not necessarily sure. But then again, the labs are kind of controlled chaos, so you don't necessarily know what's going on.
Yeah.
Um, so
What do
yeah,
we got
next one
next?
is adult only. So we've got um uh
Oh, giving it a much
yeah
later.
much later in the F2 generation.
in the F2
So these are like
generation.
looking the grandsons or daughters of the
Yeah.
previous generations. Um,
But again, this shows us it's that it does hit this transient thing. Like the generation it's given is the generation it has the effect.
yeah. Uh, Yeah, and this next one is also looking at hmm, da, 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 so yeah, again, this is
E.
yep. E
So E, they're giving it in all generations up into F2, and then I mean, it has the effect all the way through. I wrote that this is like reinforcing 5E.
yeah. Yes, that's right. And
Oh
yeah.
yeah. It is just it's all about this idea of like the generation that you give it, it yeah it has the effect of not letting it pass to the next, not letting it work in that one generation.
Yeah. And then we get our basic model of what's going on. So it ingests P14 and P11, and that gets passed on somehow and enters into like it gets
Well, it has to. It gets past. It definitely has to. That RNA has to get through the
yeah,
intestine into the body in some way. Yeah.
and then it has creates some sort of effect that is maintained by PRG1, uh, and that leads and then like SO1 capsids go into the germline and soma, and then that behavior is replicated in the later generation.
Hmm.
So now this is where they're trying it on all sorts of different strains of C. elegans because there are lots of them out there. There are lots of different, and they have different mutations. So the question is,
I mean, it's a
yeah,
lab. It's a lab creature, right? Like, there's of course like the one reference that a lot of people use, the N2, but like there's all these different variants that exist out there.
yeah. You always have to be prepared for what because there could always be like other variations in genetic diversity they don't get in the lab because it's a very domesticated strain. Whereas in the wild,
Hmm.
they might have a lot more genetic diversity, and so they might have different behaviors in response to things. So
Yes, yeah.
yeah,
So these different strains are either they are their own inbred strains or they're like maybe they got outbred once, right? With some more wild genes.
yeah.
A more wild
So
elegans.
like they got JU 1580, and it basically exhibits the same behavior both with real Pseudomonas and just the RNA, and that's transmissible between generations.
I think the one thing to say that's important about this is this JU 1580, it has a natural insertion into SIR1, or it's in a different
Yeah,
location, something like that.
that's right. So one is in a different location. So if the so one's location was
Yeah.
having an effect, then we'd see that difference. But it seems like we just need the intact
Yeah.
retrotransposon somewhere.
Hmm.
And
Hmm.
yeah, they find that it also gets like inherited between different generations. Um,
Yep. It's the
yeah,
same. Where the SIR1 is in the genome doesn't matter. Uh-huh.
yeah. Whereas in contrast, we've got this CB4856, also known as a Hawaiian variant, um, which doesn't have this effect. And the interesting about this one is it doesn't have SIR1.
Yes.
So what? And this is just a panel of different C. elegans, which have the SIR like
Which
viruses.
some have SIR1 and some don't.
Yeah, some have lots, like so N2 has lots, and other ones they just don't have very much at all.
Mm-hmm.
And so here's like a big panel looking at whether all these have that kind of preference, and
Right.
it's basically how you'd expect. We're looking at that. So
Yeah.
the more so one you have,
And I think the summary is in the next
yeah,
slide, perhaps. Yeah.
yeah. So right, we've got so essentially like once you like eliminate SIR1, you eliminate all that P11 induced learning. So
Mm-hmm.
I think
Mm-hmm.
that's
Yeah, so like SIR1 by IF, that's all. So they only have one example, right? The
mm,
ED3040, where it has SIR in the genome. It makes SIR in, like, when you look at the bodies, there's SIR1 capsids, but it doesn't have it doesn't have the learning.
yeah,
But that's because it has some other behavioral strange things in its chemosensory attraction, normal attraction,
right.
some other behavioral
Yeah,
defects.
so like if it doesn't have like if it DAF7 neurons aren't behaving how you'd expect, then it might not have that same level of attraction, might not behave differently. So
Yeah, I don't actually remember what that assay is. Like, I'm not sure if it's specific for DAF7. I think it's like some other. I don't know how they measure normal attraction, is what I'm
right,
saying.
okay. I assume that they
I, I, don't-
yeah. Yeah, you're right.
I don't want to attach it necessarily to that DAF7 because, again, DAF7 was just in that transgenerational one, right? Like, it gets turned on there, but there are many other genes that can just stop them from even learning preference in P0.
Yeah.
Yeah, so I'm assuming it's some sort of other lesion like that would express itself at P0. Yeah.
Yeah. Uh yeah, so now we've got that whole panel of organisms showing
Yeah.
what we,
So, like, I mean, to me, what this data kind of throws into the mix is, like, I love thinking about the perspective of the organism that we're
mhm.
studying, right? So, like, what is it doing for C. elegans as a whole, right? And it can't be like it's so specific, actually, right? Like, it's only like a regional. It's like only in some C. elegans does this do these genetic factors come into play. So it's not like a species wide adaptation, but rather very specific for species that live in certain
Right.
scenarios. What those scenarios are, like, I don't really know because I don't know how all these lab strains map out to what people think, like, because C. elegans is also just one species of nematode worm, right? Like, there's many species of nematode worms out there. And so I guess there is a question whether or not we think that this story about adaptation mediated by viral like particles, is that a story that we can tell about all nematode worms, or is it like some, like, just like a weird thing that we learned in the lab?
Yeah, exactly. So yeah, this one is just looking at the P11. So this is the same experiment as we've seen before, but with P11 instead of the learn phenotype. Then we have J where
Yeah.
they look at SIR1 necessity in KR314, like it's ascertained by knockdown. Um, so yeah, that
Yeah, so just to show that for other strains that have active SIR1, if you knock it out or if you knock it down, it removes that effect of the choice index in the next generation.
and that brings us to our final figure, which summarizes pretty much all of what they're trying to say here. That yeah, so that it can be passed down through generations. It can also be passed down via worm death and lysis. So if a worm dies and is killed by the Pseudomonas, it's going to explode and it's going to leach all these SIR1 capsids that can be taken up by a naive worm, and then it can learn the avoidance without having to actually encounter the Pseudomonas.
Yeah, because that's like the idea. That's as if it, like, the lysate
Yeah.
training, right? Like, that's essentially lysate training that they're saying. That they're folding that idea, that scenario that happened in the lab, into something that might be adaptive for C. elegans. That if C. elegans was browsing around a place where there was a lot of worm death, right, it might pick up this thing and then learn to avoid it.
Yeah, so I think that brings us to the end of this paper, and well, I just want to think about the mission they asked at the beginning is does Swamp Thing can be real?
Yeah, that's what the authors want.
Clearly, we want to confirm
suggest.
Alan Moore et al. Anatomy lesson, something issue 21. I don't know. But yeah, but I think that there was initial for this sort of research. There can be a lot of excitement in terms of like what this means for other organisms. But I feel like this is a very specific system for C. elegans. And yeah.
And also, like in the Swamp Thing story, that's definitely declarative memory, right? I think in some ways I tried to already debunk that a little bit, like before we went into like reading this paper through and through.
So here's me rebunking it.
Yeah, yeah. Because like this memory is this non-declarative style, right? Like it's about like experiences that shape your behavior in the future, not necessarily like what you remember, what you can recall, right? And then relate to somebody about the scenario of the past.
Yeah, and this is very much like a non-declarative memory. It operates in a very different way from how we'd imagine memory to work. Yeah.
But I do wonder, like, again, I'm not we're not neurobiologists. This is like just from me reading. It was something that you linked, Fuzz, in the document about memory. And like, I do wonder though, like, but is there interplay between the non-declarative and the declarative systems, right? Because in that example that I gave, and again, not having an expert here is doing us a real disservice, but like in that example where it's like, you know, you might not explicitly say that you don't go back to this restaurant because it has bad service. It's just like, you know, it's like, it was not a good experience. You don't want to go back there. I don't like, to me, there's like an interplay there of declarative and non-declarative, right? Because like if you were inquired in the right way, you may come up with a reason why you don't go back there, right? But the way that it influences your behavior is just kind of like in the background.
Yeah. I mean, this. Yeah.
So I don't know whether there is interplay, because then maybe there is something, right? Like maybe these are like somehow life experience, right? Like we just don't have that idea of the consciousness element here with worms.
Yeah, and that's a really big kind of question to ask. I mean, with this one, I'm kind of I'm almost wondering like what the difference is between like memory and like a passed on reflex, because this seems to be almost happening outside of what we think of as the brain of these worms.
Well, I don't know if they have, like, they don't have the same, right? They're only those 300 neurons organized in different ways. Yeah, this is they only pin down the phenomena of the ASI neuron. Like that's just, yeah, that's the one thing they pin down. But there could be other neurons, right, that light up in a. But maybe ASI was easier to, DAF7 was easier to narrow down on because it's such like a isolated thing, right? Like it only happens in that particular neuron. So like this signal on or off is a great way to tie back the behavior to a molecular signal in the nervous system. But there could be other signals, but they're being modulated by other things, right? Like neurons are getting innervated from different regions. So maybe there is another signal, but the experiment's not controlled enough for some reason to be able to isolate it.
Yeah, I think that's right. I mean, oh gosh, I had a thought about oh god, it's oh no, I remember what I was thinking about like we're looking at this from very much the worm's perspective, but from the perspective of the bacteria, being able to manipulate the worm's behavior in a way that could force it to avoid it is quite a useful trait to have potentially.
Yeah, absolutely. Yeah, like this. Right. This is, yeah, this is, yeah, from the worm's perspective, there's that. But you said that earlier, right? When I brought up, like, why would Pseudomonas care, right, to have homology in one of its small RNAs to something in a mammalian host? But yeah, if it's like Pseudomonas does live in groups, right? Bacteria do also live in groups. So, like, maybe there's a little colony and it doesn't want to be feasted on 100%. Yeah.
Yeah. But it's an interesting interplay of different behaviors, and this kind of research is like very challenging because I mean, for me, I hate having to study behavior in my normal life, so it's in a paper, just.
Yeah, and it's like they also try to sort of bring out this thought of like, does it happen in other worms, like later in the paper? And I think that's sort of that affects the way that you view this finding as well, right? Like, because if it's not generalizable to worms in whole, you have to ask yourself the question, why does it happen in some worms and not others? Like, is it about like the environment that those other worms grew up? Is it just like random chance, right? Like, there's a lot of follow-up questions that help you contextualize this one finding.
Yeah, and another follow-up is that we don't actually necessarily know what's in the SIR1 capsids. We don't know what the message is being conveyed and how that is. So there's
and how that matriculates down the whole system, right? Like, we know that without SIR1, you don't get the increase of DAF7. But what is the right? Like, what in SIR1 is giving you that increase in DAF7?
Yeah, and we still don't.
They don't sequence the bunch of
and we also don't know what's happening with those 5 generations. Why is it that after the 5th generation you don't see anything? Why does this response disappear?
Yeah, absolutely. And also this idea of like, we also don't really know like SIR uptake style, right? Like, they say when you feed the lysate to worms, right, it prevents them from going in. But like, how does this SIR1 get processed inside of the gut of the worm?
Yeah.
Right. Like, is that dependent on? Or maybe they already know. I'm not sure if they ever did that. Like, is that dependent on the double-strand transporter in any way?
Yeah. Like, there's lots of different ways. So could you like say create SIR1 capsids by like seeing the genes, creating the capsids ex vivo, and then adding them and generating this behavior without having to have any, or like do you need like, and like if we're gonna, so there's even more like kind of narrowing down. So we did have that plot where they took a bunch of like SIR1 capsids and check whether there's RNA in them. There's further research you can do along that lines where you could sequence a whole bunch of those RNAs and then test like one by one which ones.
Then make the RNAs and see what, in some ways, like fractionate even further, right? Like, they could try to fractionate even
figure out which RNAs are the ones that are prompting this response, and it'd be interesting to see whether they could prompt other responses if they're operating through this staph neuron.
Fraction 6 induces transgenerational memory. I'm like looking at the old, going back and just seeing like what thing could be open there. But yeah, I think these are all viable other paths. And I mean, if other people have thoughts about it, this is the forum to ask it, I guess. Write in our comments, right? Like, any open questions. Do you have anything else you want to say, Faz?
No, I think that's everything.
Okay, then in that case, join us next week for another news week where we're going to survey a bunch of papers and find something to cover, give us like our 2 cents.
Yeah. So if you've made it to this far, thank you so much. Please like subscribe, and if you have any comments, or because we're not neuroscientists, we're just microbiologists with opinions, and because we want to hear from you, so chime in via our Twitter or our comments. And yeah, it's been an absolute pleasure having this conversation with you, Danny.
Same here, Fuzz.
Oh, sorry. Your internet's murdering us again.
Oh, really?
Hello?
Oh, no.
Yeah, I'm sorry. It's a New York marathon, I presume.
Yeah, probably. I do hear cheering outside. Am I kind of back-ish?
Yeah, you're back. Okay.
Nice. Yeah. We want to remind everyone that we're very enthusiastic with microbiology and like somewhat qualified, especially this one. We're like stepping outside to like try to bring in all this neurology. So it's possible we didn't get everything right. If you have any questions or corrections, just let us know in the comments.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Bye.